I will present this research, which is in fact a series of articles. So, majority of you have already heard the beginning of it in Ceuta and in Barcelona, but the thing is getting with each year sharper, and some new findings are here to be presented. So I'll just quickly go through everything. So maybe the beginning you already heard, but some of you didn't, so you have a continuity. I will explain the thing. Okay, we are all here because of Iboga. I would like to point out just these indications for use, just to remember, because at the end of the presentation I'll try to bring it together to present a common denominator of all the actions at these seemingly diverse purposes of use. What do they have in common, like stimulant, ritual substance, reconvalescence after disease, religious use as a representative, as well as traditional uses, as well as urban traditional use, religious use, seen also in the West, like with Sacrament of Transition. And what do they have in common? How can we explain with a single theory reduction of abstinence syndrome, acceleration of detoxification, and the tolerance reversion, which is normally absent with explanation, because it's not the same. Detoxification is elimination of drug from body, which can be accelerated with sauna or with the diuretics, a lot of drinking. But that's not the same as tolerance reversion. Tolerance reversion is a process where body annulates its tolerance, its use to drugs. Normally it goes from somebody who starts with drugs, changes his metabolism from physiological to certain degree of pathophysiological, which is in fact tolerance development, and to go back it takes time, and it takes effort, will, and also, as we will see, energy. Elimination of cravings is another aspect which is more psychological, well explained with the neural circuits, so well documented that we start to think that the addiction nests in brain. But as we will see, in fact, it nests in the whole body, in every cell. Each cell in the body can get used, adapted to changed milieu, which means presence of drugs, slowly adapts to it and stabilizes. And when the drugs is absent, you get a rebound effect of some kind of, it's hard to say that the cell craves, but it does on its own way. It feels leakage of drug and responds with a certain stress. So we have a acute effect which is well documented with and studied as well with a so-called dirty drug, not like morally dirty as we all know, but it's because of it acts on so diverse types of receptors, transporters, enzymes. But what we see with Iboga is escaping of the normal pharmacological logic of action as long as the substance is present. We see with Iboga that we have a prolonged action afterwards when the Ibogaine is already eliminated from body. And even this effect goes far beyond the elimination of noribogaine as well, which indeed is more lipophilic. It traps in a fat, gets trapped in brain. But anyway, we cannot explain such a prolonged action in terms of months, years, maybe even life-changing experience just with this pharmacological two-compartmental system. So this half-life metabolite, long half-life metabolite exists, but it does not explain everything. And then we see in literature that neurotrophin synthesis and release, this is the step. Neurotrophin is it affects growth and growth and survival of neurons. And here we already see that it affects structural changes, intracellular connections with dendrites, and also it supports survival of cells. That means it affects also intracellular, subcellular level. So this long action of the pest, long effect can be, of course, needs to have certain hardware substrate, so to say. It's not only the psychological aspect of the experience of that we know a lot about, but I won't talk so much about this part. But we expect certain functional and structural remodeling of brain and the subcellular level of cells as well. So the basis of this cannot be others than gene expression and protein changes, which are the DNA and proteins are in fact the carriers of life, the material carriers of life. So if we look into the proteins, which are the carriers of function in the cell, DNA is only information which is translated into proteins. We can divide these proteins into two main groups. One are structural, which carry the morphology of cell, also intracellular space. We all know collagen and actin in muscles, and also cytoskeleton. It looks like cell looks like this. These proteins are high abundance. They are a majority of proteins in cells. But on the other hand, we have a minor fraction, low abundance fraction, but very important because these are functional proteins that carry the function of the cells. So these are enzymes, receptors, transporters, and also others. So we expected certain changes in the protein level, so we used this. We use this two-dimensional electrophoresis. It's a methodology of separation of... Bob, sorry, I find this very disturbing. Could you leave that for later, please? Thank you very much. So this two-dimensional electrophoresis is a method of separation of proteins. On the left side, it's a theoretical thing, and on the right side, you see how these gels, they're transparent, and you release proteins through it under the electrical current. So you divide it by their molecular weight. That means how big they are. And left, right, you divide it by how they are charged negatively or positively. So the trick is like with all the science approach, you have a control group and you have a treated group. And then you can compare the two. And if you notice the change, then you see that somewhere something has happened. It is very hard for an eye to do that, so that's why the certain softwares are developed which help to differentiate between, we see a certain spot, we notice that on the treated sample it's raised as a hill, you see it's a bigger. So then we get a quantification of these changes.
Sorry, last question. These proteins, are they isolated from certain cells of what species? I mean, is it rabbit, is it human brain? I mean,
this is a theory, yeah, we'll come to that.
Oh, so you haven't done this experiment, or you have done this experiment?
Yeah, I have done this experiment.
But on what species, or what's
We'll come to that.
Okay, sure.
Yeah. So when you see that a certain spot differentiates from the control, so it can be increased or decreased, you cut it out, analyze it with mass spectrometry. That means you identify to know which one, what is it about. So we did we treated rats with Ibogaine with 20 milligrams per kilo intraperitoneally, which is similar to oral ingestion. And we sacrificed one group at 24 hours and another at 72. It means day after the treatment and 3 days after the treatment. And so we found that we get raised levels of certain enzymes which all fall into the group of energy metabolism related enzymes. It's a cluster which is we all species, all eukaryotic, that means plants, fungus, and animals, we share practically the same. It's so-called housekeeping or basic metabolism because it's a central pathway which serves to provide energy for other functions that are more specific to the species or tissue, like, let's say, keratin production for skin cells, etc. So we see that this induction it took some time to develop. You see there that after 24 hours we have a certain raise, but the most pronounced is 3 days afterwards. So this process takes time. These enzymes are in this chain of metabolism of glucoses, where sugar are, so to say, burned to the carbon dioxide and water and produce the ATP molecules, which are the direct carriers of energy that can enter the other processes, synthetic processes in the cells, so they fuel, in fact, all these processes. So that was our first publication, and we didn't know exactly how to do. We had to go further, so we had to change a model, because also I tried to stick to a small group of rats as possible, so it was the lower, the 3 and 3 for each time, so altogether 12, but I decided then not to go on with it. And there are also other causes, to be honest. It's cheaper to take a yeast cell, which is in science a accepted model for study of primary basic metabolism. It divides quickly, it's easy to handle, it's grouped reproducibility, etc. So first experiment was to see if we get the same induction in yeast. Since it has a faster metabolic turnover, we took 5 hours after the treatment with 1 milligram per liter in media, which represents, in fact, the concentration in brain after 20 milligrams, approximately, after 20 milligrams per kilo. So we tried to make the same conditions, and what we found out was even more pronounced effect of Ibogaine on the same cluster, not exactly the same enzymes, but on the same line of these enzymes in glycolysis. So there comes a question, why this happens? Is this some kind of response to a certain event that happens in the cell, or is it just some effect of Ibogaine that it would be direct? Because it's possible that some substances act directly on the promoter regions of genes to stimulate the expression of those genes. All cells in the body carry the same set of genes, but they are not the same. The pattern of activity is not the same. So certain hormones wake up certain genes, certain substances. It's so-called elicitor. So Ibogaine could be elicitor directly for these enzymes, or this induction can be a response to a need. So we measured the level of these ATP molecules after the Ibogaine to see if it's raised, that means that Ibogaine directly affects a genome to raise this synthesis. If it falls, that means that this is the first event is a fall of ATP, and the induction is a response to it. And here you can see a dose-dependent fall in ATP pool. The pool means the quantity of these enzymes. It's a level of energy of cell. Acutely, that means in the first hour after the Ibogaine. So this induction of enzymes follows, is a response, adaptation to this transient fall. So another question comes. What happened? Why this fall? What causes this fall? Is it bigger consumption or lower production? Because the level is a dynamic balance. It changes all the time in the cell. If you go to the gym, you consume energy, ATP falls, and then you get rebound effect, the same as an exercise, induces these enzymes, so you get a bigger production. So what we did, we measured the production itself of ATP. It's hard to measure it directly, but we measured it indirectly by the production of carbon dioxide, which is the end product of glycolysis. So you see that in the first hour, we got again dose-dependent raise in ATP production, which is also a bit interesting because it repeats at a point of 3 hours later, and then everything calms down even under the level of the control. So we got an answer, because in spite of the bigger production of ATP, at a certain moment we have a lower level of ATP in the cell. So that proves that there is a consumption of energy in the cell after the Ibogaine. Acutely greater consumption of ATP. So who is the consumer of this ATP? We checked for a possible toxicity, which was at those concentrations was negated. There is no certain expenditure about transportation of Ibogaine into or outside the cell, which sometimes represents certain energy load, because Ibogaine is lipophilic and transpasses simply through the membrane. So the last thing that stayed was the possibility was to go back to analyze even more profoundly these proteome changes that we noticed in the beginning. And when we went also in this small fraction of functional proteins, we found out that it's non-specific activation of this functional fraction of proteins. We don't have yet identified all of them, and we don't have complete answer to this size systems biology story of it, all the effects of Ibogaine. But part of it is the synthesis of the energy enzymes itself. So here you see that 75, the ratio moves towards the functional fraction. And this, in terms, in a language of computer, of systems biology, represents something that we call shift in epigenetic landscape. Now we are in era of epigenetics. That means that these are changes that are not genetics. We don't have a mutation, or we don't put in the cell some new gene or take it out, but we can influence the expression of certain genes. We change the pattern of expression. And this is a mathematical model that represents a movement in such a land, epigenetical land, which is an illustration of what is going on. The concept is not new. In the 60s, Waddington first drew his idea how the undifferentiated cell can take different paths in its differentiation. You see that ball that can go by different volleys down. So today we understand that metabolism, homeostasis, is not just one right position, but we have a numerous possible positions, but each has its own energy demands. So one are more stable, one are more economic, and the others are uneconomic. And here we see that some pathological conditions, one of them is also addiction, is very energetically, very economically, in all levels, very demanding and stressful and destroying. And so how to bring somebody who is it's my personal illustration, in this valley of tears of addiction, how can we help somebody to get out metabolically, to detoxify, to reverse the tolerance, which means that you have to go from one valley to another one, more sunny, more healthy. In between there is a hill, there is a bridge. And Ibogaine attracts somehow metabolism from one position to another. But what we have in between is a certain period of effort. We have to put some effort. It costs energy. And this is, if we illustrate this ATP pool as a niveau, as a level of water in this box, you see it's a dynamic production and consumption. And we see that this synthesis of enzymes to move, when we move in the epigenetic landscape, consumes a lot of energy for a period of time. And afterwards, these energy enzymes are cytoprotective, bring more flux into another box, and we raise the energy. What we have an acute effect of energy denivellation, which is now very important to understand that process during the Ibogaine, even though considered as a less hard for an individual psychologically, for him to detoxify himself, is metabolically very heavy loading. And if a person is already low in energy, which is often a case with a pathology, with a serious addiction, or if this shift is big, that means we go from high dose of methadone to detoxify, to reverse the tolerance of high dose of methadone, then this cost, energy cost, is so big that sometimes overcomes the energy buffering capacity of the cell and the body and can result in complication. So who will be first to starve? The energy most demanding tissues. The energy most demanding tissue in mammals is brain cortex, the first one, and right away the second one is the heart. So with the brain cortex, we see one interesting effect which we all appreciate. Its activity is suppressed because of leakage of energy, so we get archaic contents of the lower, older parts of brain to come in the first place. But on the other hand, we have heart, which after each heartbeat needs to repolyze. This is a period of QT, and if we don't have ATP, who pumps the potassium in the cell to repolyze, we have the prolongation of this interval, which can result in heart arrhythmia. So, on one hand, it would be a protective to give a bigger. Here we have magnesium. He's a cofactor at energy and enzymes, a few of them in that line. Magnesium is good. Sugar, glucose, would be good. But on the other hand, can reduce the experience of a user. And we see that in some cultures, if the experience is too intense, they give a sugar cane to suck to a person because sugar lowers the effect. On the other hand, post-starvation, prior starvation, raises the effect. Not just because of higher absorption from the stomach, but if you are low with glucose in the blood, then this energy leakage in the brain cortex is even higher and the effect is even more pronounced. We have other techniques also to non-chemical, like breathings, that also can suppress activity of brain cortex. Trance is also one of them, but the mechanism is a little different. But it's very important to understand that we have here, even though the detoxification is like easier, it's easier because we have acute effect on the receptors, on opiate receptors, on NMDA, which is a principle of dissociative anesthetic, and the person doesn't suffer, but the body suffers a shortage of energy. So my personal recommendation is to go prior detoxification, and then Ibogaine as a consolidation of the last picture that you have, which is very protective against recidive, against craving, because long chronic use of drugs do have a choking effect on energy metabolism. Let's see alcohol. Alcohol is a direct source of energy, and there's a bunch of articles also dealing with methadone, with cocaine. They all influence the energy metabolism. They give it instantly. They give you energy, and on a long term, they take you. They suppress this glycolytic pathway. So if Ibogaine can change this, can reverse this, then we have biochemical basis of getting somebody away from the state where he feels, he recognizes a desire, a wish to take drugs. And it's, I can quote William Burroughs. You know, it's often people say, I feel like ran out of gas, I couldn't stand up, abstinence syndrome or opiates. It's an energy shortage. But if you give him, it's not just that a drug goes from the body, let's say it's the second, the third day after the last dose. You put then Ibogaine to a person that is very low in energy already. So it's a question, isn't it more dangerous, the abstinence syndrome, than the presence of drug in the body? So I would go on a prior detoxification. I know that certain people are not capable of that. We have to decide that's another ethical question now, how to work It's safer for a provider, it's safer for a person, but it's true that some people are not capable of detoxifying in advance, and maybe they will eventually die because of drug, because they didn't take ibogaine. That argument stands. But for me personally, I think the proper time for ibogaine for long-term success is when somebody is capable of getting off the drugs, not by himself at home. He can get with a provider if he has facilities, but they can take them off the drugs. That, of course, is a big question of high methadone doses and so. And at last but not least, it's hard to sell this kind of philosophy. A lot of addicts say, so why do I need then ibogaine? But the older ones know that it's not so hard to get off than to stay off. And I see here the potential of ibogaine.
Roman, can I ask you a question?
Yeah.
How long would you recommend?
When the first night that he wakes up and he said, I really had a good sleep, I'm rested. That means that his energy level is okay. But that's from
Weeks or months, right?
whatever. With some 2 days, 3 days, with some, yeah. So here I can also show you, we measured this oxidative stress, which is also a kind of indicator of stress itself in wider term. We see that 1 milligram per liter, and this one is just after 2 hours after the administration to yeast, which means it's the end of the acute phase. We see that we have this positive effect with the low dose, but higher dose can overcome 20 milligrams per liter. We see that it already is above the control level. So here starts the oxidative stress for a normal body. I don't know, we couldn't make yeast, we are not so far to make yeast addict. I mean, but yeah, eventually we'll go in that direction. But I guess yeast also can get some tolerance to it. It's biochemistry. It's a complex system, You pull here, it goes. It's a multi-dimensional mathematical system, and if you press here, everything can. It's a principle of butterfly and chaos. You press here and something changes elsewhere. That is epigenetic landscape. Even though we saw that after 5 hours we had half lower the oxidative low ratio with 20 milligrams. But it's necessary to divide between acute and prolonged effect. On the long term, everything is better than metabolism is capable of sustaining higher stress after the ibogaine treatment. So maybe a hypothesis is to give ibogaine treatment on drugs and prepare to detoxify later. But that's, it's just a piece of hypothesis. I wouldn't go that direction, no. Ibogaine is adaptogen. Adaptogen is a new term of a new group of pharmacological name for substances already recognized with Food and Drugs Administration and also with the European EMU, being invented in Russia. In fact, 4 substances that, like ginseng, that look like stimulant but do not have rebound effect that you would like caffeine, drink a coffee, and after an hour you are paying back the debt, paying back the rates, but helps to adapt to a stress in a manner that without harmful effect, you are able to sustain a higher stress without risking a disease. And it looks like that ibogaine suits in that group now. There is another quotation of Castaneda that maybe not so much in context of anti-addiction treatments, but maybe more in a psychospiritual way. He says that it's not about the shamanism is not about knowing certain things, but how to save energy. This energy part of the story with an old pharmacology of receptor things, we forget about its importance. If we look at global politics, we see that it's all about the energy and oil. So why wouldn't it be also on a microscopic level? So what is the outcome of this increased energy availability that we have after such a turn? This turn, when this turn is accomplished, we have a greater energy availability, and we have enhanced mental agility, elevates mood, life will. During the treatment, it facilitates introspection. This is this acute phase to have a certain psychotherapeutic insights, but in a long term, it eliminates craving with a higher level of energy and protects against the recidive. So this study showed that Ibogaine influence on metabolism is neither species nor tissue specific. It is not mediated by binding of Ibogaine to receptors that are extensively described in literature that we have, because yeast does not possess those receptors. And Ibogaine shows remedial effect in different pathological conditions, not just addiction, while in health helps to resist stress in an adaptogene manner. It can serve as a doping agent. It can be as a, we see also other uses as a relevation thing, spiritual thing. They all have they all root in energy metabolism. So this, if that's it.